welcome to CBSE NCERT Solution. I am your own biology master teacher, Miss Gopika. So today, as I promised you yesterday that we finish biological classification and I'll be starting the next chapter, that is Animal Kingdom. I hope all of you are excited for Animal Kingdom. So what we are doing in this series is NCERT line by line. Okay. Now what is NCERT line by line, ma'am? I have never heard of this and I'm a 12th student. Now this is the kind of questions that will be in your head, or you might also be a 11th student. Now what we are exactly doing is we are going to take snips of NCERT and explain every single line of NCERT and give you a recap or summary for that chapter. Okay, now it's very oftenly done, but it is not done in colleges, it is not done in places, coaching centers, because it takes a lot of time, right? Going through every line of NCERT is not an easy task, and to make every single thing properly explained is another task by itself. So we at Vedantu have taken this decision to give you this free content where we go line by line of NCERT to make you understand every single thing that you might not have understood in the college because you were having fun or you were talking to your friends, anything for that matter. Sometimes college things happen like this, so it's very normal. Now to give you that free content, we are here. So all you have to do is subscribe to the channel and also smash the like button so that we know that you love these classes. Do not be hesitant to mark your attendance by smashing the like button, because understand this is free content and all you have to do is shower some love on us. So students, before I dive into the class, let me also check if my voice is fine and if everything else is also fine. Okay, then we'll directly go into the class. Yes, we are live. I just want to check my voice as well. There's so many ads, right? There's so many ads now. Yes. Okay. Yes, we are live. I just want to check my voice as well. Very good. It's perfectly clear and audible. Okay. Yes. So let's directly dive into the class Animal Kingdom. I hope all of you are excited and let's learn Animal Kingdom. Students, now I'll tell you what you need to take is a pen and paper. Okay, take a pen and paper and keep writing what I'm telling, because in Animal Kingdom the questions that are going to come is they are going to give you an organism and they will ask you to identify the characteristics, or you have to know the phylums and much more like that. Okay, so that is your main task for the day. So you should be conscious to make note of everything that I'm telling you and also check with your NCERT then and there. Okay, ready? I'm super duper excited for the class as always. Now let's start Animal Kingdom. Now, first ever thing about Animal Kingdom. So Animal Kingdom, see, it's a very simple thing that there are millions of animals and there is millions of flora and fauna, right? Flora and fauna. Now, if imagine if like biological classification is a chapter that explained us why classification is necessary, yes or no? That is, imagine millions of animals are there and we do not have a classification. We do not know where to put them, what to do with them. We do not know anything, right? Okay, I'm starting a college and I'm not even making a categorization. Ki, okay, commerce will be there, arts will be there, science students will be there. Now, I have not made that classification at all. Now, what is going to happen? A college is going to be, okay, college is going to be like a jungle. Okay, college is going to be like a jungle. I don't think voice is low, dear. Please check your this one. Okay, commerce will be, see, voice is very clear for me. Please use the headphones so that you get a better clarity on the voice. Okay, because I'm doing it in a closed room, so that much voice only can come. Okay, the more voice cannot come. Okay, yes, just a girl, please check your voice. I mean, check the, please use the headphones to get a better clarity of the class. Okay, yes, 1 second, students. So flora and fauna, that means we have to do classification, right? We have to do classification and that classification is going to be different. Okay, 1 second. Yes. Okay, so that flora and fauna had to be categorized, right? Categorized and put into a classification. That's when we learn biological classification. Now, tell me all of you, R.H. Whitaker was the person who was like, wait a minute, there's so much of confusion. I will put all the confusion to an end. But remember, Plantae and Animalia did not start by Whitaker, right? Plantae and Animalia classification came from Aristotle. No, that plants and animals categorization I will do based on if they have RBC, if they have, depending on their habitat and all that, right? But R.H. Whittaker did what? R.H. Whittaker did a different perspective where people were finding it difficult to put fungi into a place. He worked on that. He was like, wait a minute, I know where to put fungi. Let's give them a separate kingdom. But Plantae and Animalia did not start there. Plantae and Animalia was the first ever classification, first ever scientific classification to come into existence. Before that, there was classification based on food, shelter, and clothes. That was not at all a scientific classification, right? Then from Aristotle, we got Carl Linnaeus. Again, he also did plants and animal classification. So it was like a very basic thing. Okay, plants are there, animals are there. I know it, right? Now, look here. Basis of classification. Let's use highlighter. Okay, basis of classification in spite of the differences in structure. Okay, in spite of the differences in structures and form of different animals, there are fundamental features commonly to various individuals in relation to arrangement of cells, body symmetry, nature of coelom, patterns of digestive system, circulatory system, reproductive system. So what they are trying to say, they are trying to say that in this chapter, okay, in this chapter you guys are going to study all these classifications. Okay, pen is too thin, no? Yeah, okay. Oh no. Yeah. So they are trying to say that you are going to study cells. You are going to study if they are radially symmetrical, bilaterally symmetrical, right? You are going to study the body symmetry. You are going to study the nature of coelom, right? If they are acoelomate, okay, pseudocoelomate, and then you are going to study the patterns. You're going to study if they are, how their digestive system is, how their circulatory system is, how their reproductive system is, respiratory system is, everything about the organism you're going to study. Okay, and then we will get to know, okay, wait a minute, when zoologists or botanists were doing the classification, they had a clear picture how to categorize them, on what basis they should categorize them. Okay, I hope the first paragraph is clear now. Students, before we directly dive into the chapter, please try to understand that this chapter has a lot of terminologies, okay? And that is the reason why students find it very difficult to, cope up with this chapter, telling them, ma'am, I'm not able to remember. So what you have to know is, first you have to learn some concepts or some terminologies, okay? So we are going to first deal with some terminologies. Terminologies. See, I'm writing as much as possible, okay? As much as possible. It's not a board setup, so I have limitations to write, but I'm trying to write as much as possible. The rest, everything, I'm going to try to explain and make you understand. All you have to do is stay till the end of the session, okay? Now, see, though all the members of Animalia are multicellular, okay, all of them do not exhibit the same pattern of organization of cells. What are they trying to sell? They are telling that some of them are maybe loose aggregations of cells, some of them might be tissues, some of them might be organ, some of them might be organ system, but for the fact that we shouldn't forget, all of them are multicellular. Okay, they're telling that all of them are multicellular, right? It's very simple, students. Imagine us only. Now see, just a girl is there, I am there in the class, Roshni is there in the class, okay, Nandini is there in the class, Subhashni is there. So all are 5 different girls in the class. We know that we are all, okay, so all come into the gender of girls, but all of them have different fashion sense, all of them have different characteristics, personality, looks, right? Like that differences is there. So they are trying to say, when you think it is a multicellular organism, do not have the, okay, do not have the concept that, okay, everyone, okay, everyone are going to be the same, everyone are going to have the same cell aggregation. That is not the same thing, okay? So same pattern of organization. For example, in sponges, the cells are arranged as loose cell aggregates, okay, loose cell aggregates. That's what I told you. Some of them were loose cell aggregates, some of them were tissues, some of them were the organ system, and so on, okay? They exhibit cellular level of organization, okay? Some division of labor occur among the cells in cell traits, especially in cell traits. The arrangement of cell is more complex. Here, the cells performing the same functions are arranged into tissues. Hence, it is called as tissue level of organization. So now what you have to understand is that all the cells are multicellular, but let's use another color. They all have, okay, if I write here, it's visible, no? They have cellular level of organization, Okay, that means who have loose cell aggregates. Okay, all the cells are there. Okay, all the cells are there. Got it? Then they have tissue level of, okay, tissue level of aggregations where all the tissues are there. Okay, tissue level. Then they have organ level. Obviously all the tissues have decided ki I am going to do this function, so even you are going to do this function. So it's very simple, students. Now imagine different tissues. Okay, let's act like all these tissues can talk. So all these tissues are like, hey wait, I also do breathing, even you do breathing, even you are a part of this. Then another person will tell, you know what, even I am a part of this because I am getting the I'm transporting the gases. So all of these people, no, all of these people are together going to form the respiratory system. So division of labor has happened. That is, a certain set of cells are like, wait a minute, I am going to do this job, you going to do this job. Now example, just a girl is going to make sure that the attendance is marked in the class. I am going to make sure that I'm going to deliver the teaching in the class. Another student is making sure that, okay ma'am, ma'am has done this, I'll make sure that ma'am sends the notes in the class. So this is what division of labor. Each of us have taken one job and each of us are doing together. Okay, aggregate means association, like all of them coming together. Okay, all of them coming together and they forming a set of, it could be anything, like all of us coming together and forming an organization. Now Vedantu is a set of teachers who want to teach, like who has the love for teaching. So we have become like an aggregation. Got it? Yes or no? One second, students. Okay, so tissue level, organ level, got it? Organ level, and that's it. So here they're telling, hence called tissue level of organization. A still higher level of organization is going to be organ level of organization. Okay, exhibited by the members of Platyhelminthes and other phyla, where the tissues are grouped together to form organs, each specialized for a particular function. In animals like Annelida, Arthropoda, mollusk. Okay, so they're telling that in one phylum, actually there are organisms which are also going to show organ system level of organization. Okay, organ system level of organization. Now see, echinoderms, chordates, organs have associated to form functional systems, each system concerned with a specific physiological function. This pattern is called organ system. So here we are going to talk about organ system. Organ system. So we as so far what did we do? We understood cellular level of organization, tissue level of organization, organ level of organization, organ system. Okay, organ system where many organs come together and they form a system. Okay. Now organ system in different groups of animal exhibit various patterns of complexity. For example, digestive system in Platyhelminthes has a single opening to the outside of the body that serves both as an anus and hence it is called incomplete digestion. So students, how can I call something is complete? Okay, how can I tell that the class is complete? Because I can tell the class is completed when I start a topic, I finish that topic till the end of the class. Yes or no? Same way, if I have to tell that the digestive system, I'm giving you so many simple example. Don't be like, why this ma'am is talking so much. It is because to make you understand in the as simpler as possible. Okay, as simpler as possible. So don't be like, ma'am, why are you telling so many things? Why are you telling so many things that we are not, don't tell like this? But actually understand that many students find it very easy to relate with normal day to day things. Okay, yes. So digestive system, if I have to tell it is complete, okay, if I have to tell it is complete, I will tell that it should have a mouth. Okay, it should have a mouth and it should have an anus. It should have a mouth, it has should have an anus. That means two openings should be there. Now when do I tell it is incomplete? When do I tell it is incomplete? I'll tell it is incomplete when there is only one opening. One opening. That's all. That's when I tell it it's incomplete. Basically, there were supposed to be two openings, but when only one opening is there, that is when I will tell it incomplete. Is this clear? Yes or no? Let me also check the chats. Okay. Okay. 1 second, students. Yes, the chats are moving now. Okay. Yes, clear. Okay, done. Very good. Okay. Yes, ma'am. Fine. A complete digestive system has two opening, mouth and the anus. Similarly, the circulatory system may be of two types. That is open circulatory system and closed circulatory system. Now we have discussed a very slight topic of this. We would have discussed in many other chapters, right? When we have spoke about, let's say, even in biological classification, small things also we have discussed this. Now students, oh sorry, open type of circulatory system means what? Very simple. Okay, blood is there. Okay, blood is carrying all the things that is required. Blood is not going to transfer something, like in the sense that transport is not happening because of small tubules like capillaries. Okay, capillaries are not there. Instead what blood is, so this, when there is capillaries and the capillaries are taking it to the organs, this will be called as a closed circulatory system. Now I am sitting in a closed room. What does it mean? All the four walls are completely packed or closed. Okay, now same thing, you imagine blood. Okay, blood opens into spaces. Okay, blood opens into spaces. What will that be called? What will that be called? Sinuses. Okay, sinuses. Okay, 1 second. Yeah, sinuses. Okay, sinuses. That is small spaces. Small spaces. Okay. Yes, I think charts are moving. Yeah. Okay, spaces. These spaces are called, students, sinuses. The spaces are called sinuses. That is, imagine that blood is flowing. Okay, open means what? Now blood is flowing. There is open pipe means what, students? Like this, the water is flowing. Water is flowing and it is going like this openly. It is, does not have a proper system or anything. Okay, proper system or anything. It is just opening and it is just going everywhere. Now imagine there are different types of organs. Okay, this is some random, I'm just drawing some random structures. Okay, there are organs like this. This organs will get bathed in this blood. Okay, bathed in this blood or covered in this blood, because it is very simple. Now my hand is there are small pebbles on my hand. I pour water into it. What is going to happen? Obviously all the pebbles are going to submerge in that water. Now same way, this in open circulatory system, always remember that the organs are going to bathe in the blood. Okay, but closed circulatory system, is it like that? Imagine a pipe is there, proper closing system is there, opening system is there. It will not happen like that. No, everything will go properly through a route. So closed circulatory system, in which the blood is circulated through a series of vessels of varying diameters, that is your arteries. So blood vessels are the reason why we have closed circulatory system. Otherwise all our organs would have bathed in blood. Okay, bathed in blood. Now this is open and closed circulatory system. So you learned, right now you learned incomplete digestion, complete digestion, closed circulatory system, open circulatory system, cellular level of organization, tissue level of organization, and organ level of organization, organ system. This is how much we have covered so far. Okay, yes, so far clear? I want all of you to like the video as well. Quickly like the video. Whoever joined now, like the video. Okay, that's a good thing. That's a guru lakshana that you give for the teacher. Okay, now let's understand symmetry. Okay, I'm so sorry for moving in. Okay, symmetry. What is symmetry, ma'am? Okay, symmetry means what? 1 second. Yes. Symmetry is animals can be categorized on basis of their symmetry. Okay, sponges are mostly asymmetrical. Now, very simple thing, students. Now, I do not have anything that is very disfigured, but if I had something, I would have shown you people. Okay, now I don't have anything near me that is so disfigured to show you people. But let's say this is the box. Okay, this is one box. Now I know that I can break this box into two parts, two equal halves. But imagine this box was in some other shape, like this or something, like this or something. There is very less chances that I'll make it into equal half. Okay, make it into equal half. So asymmetrical means what? Like some sponges are these kind of shapes. Okay, some sponges are like this. Now you tell me, how do I make it into equal half? One side is longer, one side is shorter. Now there is very less chance for me to make it into equal halves. So that is why we tell asymmetrical. That is like however I try to cut it, I will not get it into two equal halves. Okay. Now, so here see the definition also is given. That is, in any plane that passes through the center does not divide them into equal halves. Okay, whatever plane you cut, however you cut with love, you cut with hate, it is not going to form two equal halves. Okay, now when any plane passing through the central axis of the body divides the organism into two identical halves, okay, it is called as radial symmetry. That is, what are they trying to say? If something is passing through the organism in the center, okay, it's passing like this in the center. Now mine is a cross line. One second. Yes. Okay, if it is passing exactly from the central axis, it is passing in the center. This side also there is a proper half, this side also there is a proper half, then this will be called as radial symmetry. That is, they can be identical. Okay, identical halves, identical halves. Okay, identical halves. Now I can cut an apple exactly through the center and I'll get two halves. No doubt in that. That is identical halves, and that will be called as radial symmetry. So when any plane passes through the central axis of the body, divides the organism into two identical halves, it is called as radial symmetry. Now, coelentrates, ctenophores, this you will study, like when you're studying phylum, you will get to know. See, coelentrates, ctenophores and echinoderms have this kind of body plan. So who and all? CCE have this body plan: coelentrate, ctenophores and echinoderms. Animals like annelids, arthropods, where the body can be divided into identical left and right halves only in one plane. That is, students, in radial symmetry. Now imagine, even if I cut like this, I'm going to get two equal halves. Okay, but in bilateral symmetry, only in one plane. Okay, this box, I can see now this box. One second. I can cut like this. I can cut like this. I am going to get two equal halves only. This is radial symmetry. But imagine this glass. Okay, this glass. Okay, this glass also, if I cut like this and cut like this, I will still get an equal half. But just imagine this is very disfigured. Okay, now what is going to happen? This one, if I cut only in one plane, only I'm supposed to cut from this plane, I'm going to get a two equal half. That is called as bilaterally symmetrical. Okay, uh just a girl, mostly we will finish as much as phylums possible. Okay, we'll try to finish all the phylums possible. Not all the phylums, at least up to Annelida, Arthropoda. Okay, so here bilaterally symmetrical means only in one plane. Okay, only in one plane it will give me halves. Okay, only in one plane it is going to give me halves. So now you have studied some more things. See, look here, this is it. Radial symmetry means, see, any side I'm trying to cut this, all the sides I'm going to get two equal half. But look at a crab, I can only cut it like this, because if I cut them like this, I'm not going to get two equal half. You might think you will get a two equal half, but you will not get two equal half, because this region is broader, this region is thinner. So only in the one plane I'll be able to get it. This is clear or not? Yes or no? This is one of the very important questions. Bilateral symmetry and radial symmetry. Okay, any plane I'll get identical halves. What? Identical halves. Okay. Diksha. Diploblastic, triploblastic, that is animals in which the cells are arranged in two embryonic layers. Okay, one of our teachers name is also Diksha. I think you would have seen chemistry class on the same channel. Okay, and our main channel is Vedantu NEET English. So yes, okay, where we take everyday classes. So there Diksha ma'am is there. So yes, animals in which the cells are arranged in two embryonic layers. So listen carefully: diploblastic and triploblastic. Okay, now students, all of you know that there are three germ layers. I think every student should know by now that there are three germ layers. Three germ layers. Now what do I mean by three germ layers? There is ectoderm, mesoderm, and endoderm. Okay, endoderm. These three germ layers are there, and this is how you all remember, no, morula, blastula, gastrula, all the stages. So these three germ layers are there. Now, if the cells are arranged in two embryonic layers, an external ectoderm and an internal endoderm, but there is no mesoderm, then that kind of animals are called as diploblastic animals. Diplo means what? Two, right? Diplo means two. So only if two germ layers are there, one ecto is there, one endo is there, then I will call it diploblastic. Why will I call it triploblastic? Obviously when three germ layers are there. Okay. So diploblastic animals, example coelentrates. Now, an undifferentiated layer called as the mesoglia is present in between the ectoderm and endoderm. Always remember, between every single layer there should be something that is present. Okay. Now, in between ectoderm and endoderm, for these organisms, that is diploblastic organism, they have a layer called as mesoglia. Mesoglia, which is not a prominent mesoderm. It has not become a mesoderm. It's just a layer of cells. Okay, an undifferentiated layer. That's why it is called as mesoglia. So we cannot calculate it as a part of the germ layer. Okay. Now, in those in which the developing embryo, okay, those in which, one second, students. Oh. Yeah, those animals in which the developing embryo has a third germinal layer, that is a mesoderm, in between ectoderm and endoderm, are called as triploblastic animals. This is platyhelminthes to chordates. So we have very few organisms who are diploblastic. We have more organisms in triploblastic and chordates. Okay, now look here, this is the layer we were talking about. Look carefully, you'll be able to understand. See, this is ectoderm, this is endoderm. So this is the most outermost layer, this is the most innermost layer. Okay, innermost layer. Now between these two layers, can you see ectoderm is there, endoderm is there. Between this, can you see this pink color layer, which is called the mesoglia. This mesoglia is what? An undifferentiated layer. Okay, this is an undifferentiated layer. Undifferentiated. It's not fully developed, it does not become like a mesoderm, it's an undifferentiated layer. Okay, now here, look here. Here you have ectoderm, okay, then same way here you have endoderm. In between, can you see a beautiful differentiated layer, which I will call it as mesoderm, That is a beautiful differentiated layer is there, and the organism who has this, okay, this kind of a setup is called, one second student, this kind of a setup is called triploblastic. I have not even missed one diagram of NCERT, so all you have to do is stick to this, teaching, and you will be able to ace in your biology exam. Okay, especially zoology. Diploblastic, got it? So diploblastic, triploblastic. Diplo means ectoderm is there, endoderm is there, middle nothing is there, but middle nothing cannot be there because we cannot have spaces. So in between we have an undifferentiated set of cell called as the mesoglia. Okay, then you have in the other side you have an ectoderm, endoderm. Between you have one layer called as the mesoderm, which is a proper differentiated layer. Is this clear? Proper differentiated layer. Now, coelom. Now let's see what is coelom. Coelom, presence or absence of a cavity between the body wall and the gut wall, is a very important in classification. That is, students, always remember. Okay. Yes, one second. Yeah. Oh no. Things ma'am do. Yes, done. Okay. Look here, students. There is a cavity. Okay, remember like this. There is a cavity between your body wall and your gut wall. Okay. Let's say this is your body wall. Okay, this is the body wall. Chick, 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 chick. Chick, 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 chick. Okay. Then this is your gut wall. This is just to make you understand. Okay, this is not exact diagrammatic representation. Now between this there is a cavity. This one. This cavity. Okay. Now, So between your body wall and the gut wall, there is a cavity, and that cavity is lined by mesoderm. Okay, that cavity is lined by mesoderm. Now what you have to remember is, okay, this cavity can be present or absent. This cavity can be present or absent. Got it? Now see, the body cavity which is lined by mesoderm is called as coelom. Okay, the body cavity in which the lined by mesoderm is called coeloms. Animals possessing coeloms are called as coelomate. Example: annelids, mollusks, arthropods, echinoderms, hemichordata, chordata. Now in some animals the body cavity, we'll use another color. Okay, in some animals the body cavity is not lined by mesoderm. Instead the mesoderm is present in scattered pouches called as in between the ectoderm and endoderm, and these are called as pseudocoelom. Pseudo means what, students? False. See, where mesoderm has to line, there mesoderm only has to line. Now, in between this cavity where mesoderm had to line, no, instead of that, imagine the mesoderm. Okay, now imagine this is the lining. Instead of that, imagine mesoderm. Okay, mesoderm form pouches like this. Okay, form pouches like this. Okay, like this, pouches are formed. Imagine instead of mesoderm, okay, yeah. Yeah. Instead of mesoderm becoming the entire lining or lining the entire thing properly, imagine they form pouches. If they form pouches, will I call it a complete coelom? I cannot call it a proper coelom. So instead of that, I will call it pseudocoelomate. Okay, pseudocoelomate. Pseudocoelomate. That means false coelom is there. False coelom is there. Okay. Now example is Ascalminthus. The animals in which the body cavity is absent are called as acoelomate. Okay, which are absent is called as acoelomate. Example Platyhelminthus. The only one that has acoelomate one is going to be Platyhelminthus, and the only one which is going to have pseudocoelomate is going to be Ascalminthus. Done. Okay. Now we have so much to remember. Right, so much to remember. Yes, look at the diagram here. Beautiful diagram is given, students. This is the coelom. Can you see? So this, okay, this is the endoderm, this is the mesoderm, and this is your ectoderm. Okay. Now here look at it. The mesoderm has become into pouches. Kuti pouches they have formed. This still remains as the ectoderm. Okay. This remains as the endoderm, and these are the cavities. Okay. These are the cavities. So we will call it as pseudocoelom. Okay, we'll call it a pseudocoelom. Here you see proper ectoderm is there, mesoderm is there, endoderm is there. That is called as acoelomate. That is, there is no cavity only, right? How triploblastic I showed you, it is exactly like that. Where is the cavity between these people? Like this cavity should be there. No, there is no cavity only. So they are acoelomate. Okay, they are acoelomate. No coelom only, no cavity only is there. Okay, these are the three different categories. Now, next is segmentation. That is every organism. Now, all of you would have seen a earthworm, a caterpillar, and all of that. How the worms look, students? I love drawing caterpillar. Okay, not because no, I think if I draw it here, it's not seen well. See, this is how your caterpillar will look like, no? Caterpillars have to forgive for this diagram, but it's okay. It's to make you people understand. Okay, this is how your caterpillar is there. Yes or no? No? Oh, that's a sad caterpillar. Okay, 1 second, students. I can do this. Yes. Now, yes. So this is how a caterpillar is going to look. Small like this will be there. Now look at this caterpillar. Now if I give a border for the caterpillar. Okay. Now if I give a border for this caterpillar. Like this. Understand that this caterpillar shows segmentation. Yes or no? Each of the segments I'm able to understand. Yes or no? Now at the same time, if I draw an earthworm. Okay, if I draw an earthworm, earthworms are long like this. Earthworms, if I draw, students, how will the earthworms looks like this segmentation will be there, yes or no? Like this full segmentation will be there. So what is the segmentation showing? That is what here they are going to discuss. Okay, here they are going to discuss. Yes, look here. In some animals, okay, in some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called as metameric segmentation. Okay, metameric segmentation, this is what you see, metameric segmentation. That is, sometimes in some of the bodies they could be divided externally also. That is, one earthworm is there, see, this is one worm. Externally you can see the demarcations, internally also they will be divided. Okay, so they are telling in some animals the body is externally and internally divided into segments with serial repetitions. Okay, continuous repetition of at least some organs. For example, in earthworm, the body shows this pattern called as metameric segmentation. This is the earthworm which you can see, metameric segmentation, and the phenomena is called as metamerism. That means segmentation that you see. Segmentation. Got it? Okay. Now notochord, next is about notochord. Okay. Notochord is mesodermally derived rod-like structure formed on the dorsal side during the embryonic development in some animals. That is how we have now backbone, right? For that, instead of that, there is notochord that was present in the dorsal end. Dorsal end is the end which is facing the outside. Okay, ventral, dorsal. So dorsal end, there is a notochord formation. That rod-like structure was formed in the embryonic stage with the help of deriving from mesoderm. Okay. Now animals with notochord, that is animals whoever had notochord, got it? They are called as chordates, and those animals which do not form this structure are called as non-chordates. That is, the ones who had notochord in their embryonic stage later developed into vertebral column will be called as vertebrates. The ones that did not form into vertebral column will be called as non-vertebrates. That is a different story. But understand, if they ever had notochord, we will call them what? We will call them chordates, and if they did not have that is notochord, we will call them non-chordates. Okay, so chordates and non-chordates, porifera to echinodermatas. Now this is the most important classification. Yes, so far good, no? Okay, so look here, kingdom is there, animalia is there, is a multicellular organism. Now animals are divided into a cellular level of organization, tissue, organ, and organ system. Okay, now cellular level of organization mostly asymmetrical. Body or cavity or coelom will be acoelomate. Acoelomate. Okay, now acoelomate, you have only one example, that is your porifera. Okay, porifera. Now in tissue, organ system level, you have radial symmetry and bilateral symmetry. If you go to radial symmetry, you will notice that there is acoelomate organisms where you have coelenterata. In this category, only acoelomate is porifera. Okay, understood? That is mostly asymmetrical. Asymmetrical they have cellular level of organization. The only people who are present are porifera. Okay, now radial symmetry, you have acoelomate, you have two people, coelenterata and ctenophora. Okay, now bilateral symmetry, that is without a body cavity, you have platyhelminthes, which is an acoelomate again. Now with false coelom, you have only one fellow, pseudocoelomate, that is ascalminthus. Now with true coelom, you have coelomates, annelida, arthropoda, mollusca, echinodermata, hemichordata and chordata. Okay, got it? This classification, if you want, you remember. If it's too difficult, don't remember. Yes, let's do a recap of what we just understood right now. Okay, so from where do we start? So students, cellular level organization, tissue level, organ level, that you understood. After that we did symmetries. Okay, after that we did symmetries, we did triploblastic and coelomate. Okay, in the order that I remember, I'm going to write it. So first we will start with symmetries. Okay, symmetries. Okay, you can't hear. I'm going to write it. So first we will start with symmetry. I find it fine only, converge study. Voice is fine only. Okay, symmetry is going to be one portion here, one portion here. We have bilateral symmetry. Okay, bilateral symmetry. Then we have radial symmetry. Okay, radial symmetry. Radial symmetry. Now this bilateral symmetry and radial symmetry. One second, I have a actually mic is there. One second, let me just check. One second, students. I'll just get the mic. I'll just check if it is going to work. One second, students. Just bringing the mic. I'll just check with the mic once. I'm not sure if this is going to work. Let me check. One second. Just give me 1 minute. Okay, 1 second. Let's check if it's working. Am I audible? Okay, I have to check now if I'm audible or not. Am I audible, students? Okay, 1 second. Let's check if it's working. Yes. Audible? Is it better? Okay, I have to check now if I'm audible or not. Am I audible, students? Diksha, just let me know in the chat if I'm audible and is it better than the before one? Yeah, it's working, no? Yes. Is it better? Yeah, but no difference, huh? Now, what about now? I'm trying. Diksha, just let me know in the chat if I'm audible and is it better than the before one? Yeah, but no difference, huh? Now, what about now? I think it's the same, but I'm using a mic. So, I don't know what's the issue. Students, from in my phone it's actually better. I don't know. Maybe you should check with your phones once and also just use in headphones because when I made my team also check, they told me it's okay, ma'am. Okay, wait. Let me make another person also check. Just check your voice. The using a mic. This is how normally how much I use this mic only, but I don't know why today it's not. Okay, yes. So let's continue for today. Adjust. Okay, tomorrow I'll try to do something else. Okay, I'll fix it for you guys, or I'll increase the volume or something. Well, let me also check my OBS once. Okay, maximum volume, no? Okay, 1 second, I'm checking. Okay. Now let's just try. I don't know why today it's not. Okay. Hello, hello. Yeah. Hello, hello. Yeah, I think this is better. Okay. Yeah, I think this is better. Okay. Okay, I'll get this fixed, students. I can do this later. Okay, now we'll continue the class for now. I'll get back to you guys. Okay, now it's coming, right? Yeah, fine. Okay, so symmetry, bilaterally symmetrical, radial symmetry. This is the two categories that we learned. Okay, bilateral symmetry means what? If I cut them only in one plane, okay, if I cut them in only one plane, I will be able to get two identical halves or two equal halves. Okay, two identical halves or two equal halves. Now radial symmetry means what? Any plane I cut, okay, any plane I cut, I'll be getting two identical halves. Two identical halves. Yes, clear? Okay, clear? Voice is fine now? Yes or no? Little better? Radial symmetry means what? Any plane I cut. I think it's similar. Okay, similar only. I don't think there's much difference. I think I have to change the settings or something. So we will do that. I will get it done by this people. Okay, I'll get it done by this people. Okay, chalo. Fine. Yes. Okay, let's continue. It's 100, yeah. Okay, yeah. Fine. Now radial symmetry understood. Any plane, two identical half, one plane, two identical half. Okay, now if I go to the next topic, okay, let's use another color. If I go to the next topic, we spoke about diploblastic. Okay, diploblastic and triploblastic. Triploblastic. Diplo and triplo. Diplo and triploblastic means diploblastic, remember that there are three germ layers. Okay, three germ layers. Now in this three germ layers you have ecto, okay, endoderm. This is going to be diploblastic. In between this there is an undifferentiated layer called as the mesoglia. Okay, undifferentiated layer called as the mesoglia. Undifferentiated layer called as the mesoglia. Now triploblastic means what? You have ectoderm, okay, endoderm and a proper differentiated layer called as the mesoderm. Okay, proper layer, differentiated and well differentiated layer called as the mesoderm. Got it? Mesoderm. So ectoderm, mesoderm and endoderm. This is called as triploblastic and diploblastic. Okay, very easy. No, diplo means 2 layers, triplo means 3 layers. Fine. Then and remember like this, diploblastic, yeah, okay. Then we'll go to coelom. Okay, here I'll do one thing, since there's some space, I'll write about digestion also. Okay, digestion also I'll write. Okay, here students, digestion, two types will be there: complete and incomplete. Okay, complete and incomplete. Complete means they'll have two openings, that is mouth and anus. This incomplete means only one opening. Okay, one opening, that could be a mouth or anus, whatever, one opening, this will be called as incomplete. Got it? Now when I come here, same thing goes with open and closed circulatory system. Open circulatory system, CS I'm writing. Open circulatory system, what is going to happen? Blood is going to flow into sinuses or spaces. Okay, spaces are called sinuses. That is because these people have no blood vessels. Okay, so these ones have no blood vessels. Blood vessels means arteries, veins and capillaries. Then closed circulatory system means what? Closed circulatory system means blood is going to flow inside blood vessels. Okay, blood vessels, and there are no spaces, everything is going properly. Okay, everything is going properly. Next thing we learned was about coelom. Okay, next thing, which color? Yes, next thing we learned was about coelom. Coelom, that is, there is this, what is the coelom? A cavity which is present. Okay, a cavity which is present between, okay, between gut wall and body wall. Gut wall and body wall. Now if, depending on if this cavity is present or not, they are categorized into different, or what is the lining of the cavity, that also matters. That is, if the cavity is lined properly by mesoderm, okay, proper mesoderm lining is there, then it is called as, okay, if proper mesoderm lining is there, it will be called as coelomate. Coelomate. If proper lining is not there, okay, if the mesoderm, okay, or let's say, when if the mesoderm is into pouches, we'll go in order. Mesoderm is in the form of pouches, then we will call it pseudocoelomate. Okay, pseudocoelomate. Now imagine such a layer does not have any lining of mesoderm, no coelom at all, then we will call it, okay, no coelom at all, no coelom at all, we will call it acoelomate. Okay, acoelomate. Clear? Okay, coelomate, pseudocoelomate and acoelomate. Okay, now this is the recap of how much we have done so far. Now we are going to start with 1 phylum. Ready for 1 phylum? Okay, ready? Yes. Okay, chalo, let's do each phylum one by one. Before that, let me, 1 second. Anytime mic is not working, just let me know. Okay. Okay, yes. So next is phylum Porifera. So let's see what is this Porifera. Okay, phylum Porifera. Members of this phylum are commonly known as sponges. So as in, when I'm reading, I'm going to write small points here. I want you guys also to do that with your NCERT. Okay, they are mainly going to have sponges in them. They are generally marine and mostly asymmetrical. I'm going to write the most important things, marine and all that you should know by now, right? They are obviously marine. We do not see them in fresh water. So they are asymmetrical. Got it? Now, They are primitive multicellular animals and have cellular level of organization. That is what we discussed in the first, that even though they are multicellular, they might have cellular level of organization. Okay, now sponges have a water transport or a canal system. Very, very important feature. So I'll write it in a different color. They have canal system. What is canal system? You will get to know now. Okay, further they are going to tell that. Water enters through. So very simple, students. I'm going to draw a sponge here. Okay, I'm going to draw a sponge here. Okay, this is a sponge. This is the sponge. Now, can you see spaces here? I have left spaces here. Now, through these spaces, the water, okay, through these spaces, the water is going to enter inside. Through these spaces, the water is going to enter inside the organism, and these spaces are called as ostia. Now this water is going to enter into which place? Water is going to enter into a entire cavity called as the osculum. So look here, the water enters through minute pores called ostia in the body wall into a central cavity, the spongocoel, from where it goes out through the osculum. So this portion, okay, sorry, spongocoel, I told by mistake osculum. Okay, this portion where the cavity is there, this is called as spongocoel. Okay, whenever the word seal comes, you know it's a cavity, right? Spongocoel. So the water has got into this cavity and it will come out from of a bigger opening. Okay, it will come out of a bigger opening, and this bigger opening is called as this one. Okay, this one, this bigger opening, say this water will come out, and that water, okay, this opening is called as osculum. This opening is called as osculum. Clear? So the water canal system works on this way. The water, okay, H2O I'm writing here, okay, H2O. The water has entered through small pores. Okay, these pores are called as what? These pores are called as ostia. Water has entered through small pores called ostia, and they enter into a cavity. A middle may there's one full cavity, that cavity is called as spongocoel, and through that spongocoel the water will go out through the osculum. Now the pathway of water transport is helping in then what? Whenever this water is coming, some food also is coming, no? Some food also is entering inside. Okay, whatever they want to capture, that is also entering inside. So the pathway of water transport is helpful in food gathering, respiratory exchange, as well as removal of waste. Okay, so the water canal system does not just do the passage of water, but it is going to help in food gathering, it is going to help in respiration, that is for the exchange of gases, and for what? Even for excretion. Okay, that is for the removal of waste materials. Three things this water canal system is going to help. Now, choanocytes or collar cells are lined in the spongocoel. Very, very important. Very important. Now, imagine this is the corners. Okay, these are the corners. I'm trying to draw as much as possible for all of you. These corner, students, there are collar cells or choanocytes which have cilia on them. Okay, which will do what movement? Which are going to help the water to move. Okay, they are going to help. Now water has entered, no? Now this choanocytes will be like, you move. That movement will be done with the help of choanocytes. This question has come many times in your NEET as well as board. Choanocytes or collar cells line the spongocoel and the canals. Digestion is intracellular. Okay, digestion is happening inside, and the body is supported by a skeleton made up of spicules or spongin fibers. So the skeleton of this what is that? Sponge is made up of what? Spicules, spicules or spongin fibers. Okay, spongin fibers. Done? Okay, now sexes are not separate, that means sexes are in the same body. Okay, eggs and sperms are produced by the same individual. Sponges reproduce asexually by fragmentation, sexually by the formation of gametes. Fertilization is internal and development is indirect, that means they have a larval stage in between. Okay, now examples you can remember is spongilla, euspongilla, okay, euspongilla, euspongilla, okay, and cycle. You can remember these two examples to remember this. Okay, There's a trick actually. Sonu eats sweets is the trick. Okay, Sonu eats sweets. So spongula, E for euspongula, and S for cycle. Sonu eats sweets. Now this is what we learned about porifera. Now what is the most important things about porifera is what we are going to learn. Okay. Now look here, students. These are the sponges. You can see this. What are they? Cycon, euspongia, and spongula. Oh, euspongia and spongula. Okay. I made a, what is that, hybrid of it, spongilla plus euspongilla. Okay, so here I'm going to write euspongia, euspongia, okay, and cycon and spongilla. So the trick is Sonu eats sweets. Sonu eats sweets. So now look here, you can look here, this is the cycon. Can you see this is the operculum, a big opening. Okay, through small pores will be there through which the water will enter. Okay, water will enter and the water will go inside and come out through this place. Okay, come out through this place. Now look here, first thing that you have to remember is about the canal system. Okay, canal system. In this, what you're going to remember, how the water is going to go. The water, okay, H2O has entered with the help of a small pore. Okay, helped with a small pore called as ostia, small opening called as ostia. Through the ostia, the water will reach into a cavity called as spongiocele. Okay, spongiocele. From the spongiocele, it will go out through a big opening called as osculum. Okay, through a osculum. Now this entire system does not just help in entering of the water, but they are also going to help with food gathering. Okay, to get food gathering, to do respiration. Okay, to do respiration, as well as also going to help with the excretion. Okay, also going to help with excretion. Got it? Now, ostia, H2O, ostia, spongiocele and osculum. Now, in the spongiocele, the most important thing that you have to remember is spongioceles have special cells. Okay, special cells called choanocytes. Okay, special cells called choanocytes which help in movement of the water molecule. Okay, like this. Choanocytes are present, or we can also call it choanocytes, or also call it as collar cells. Okay, collar cells, choanocytes or collar cells. Got it? Choanocytes or collar cells. Now what are they going to do? They are going to help in the movement of the water. The water is coming in with the food and everything. It will help them to do the movement, telling them you go, 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 go that side, right? You have seen, no, when we are going for some festival and all, they'll push us. How? That is telling them go, go that side. So choanocytes or collar cells are present. Now other some important things is that we wrote that they are going to help in food, respiration, excretion. We understood that water is entering through small pores. Okay, these are small pores called as ostia. And then you have spongiocele. Spongiocele will open into osculum, which is an opening. Okay, osculum, which is an opening. And this spongioceles have lined, they are lined with choanocytes or collar cells. Okay, choanocytes or collar cells which help in the movement. Okay, which help in the movement of the water, movement of water. Now what do we get to know? Sexes are not separate, they are not hermaphrodite, the sexes are in the same, and then sperms are eggs are formed, the fertilization will be internal, and they have a larval stage. Okay, how do you remember the example? Sonu eats sweets. Okay, Sonu eats sweet. Sonu eats sweets means what? S for cycon, E for spongia, okay, and S for spongilla. Okay, S for spongilla. Done. That is your porifera. Okay, yes, here also I've left space. So I'm writing as much as notes possible so it is easier for you people. Okay, it's easier. So students, you should always start like this, that they are marine. Next you go to their symmetry, they are asymmetrical. Okay, they are asymmetrical. Then you go to their what is that, diploblastic or coelom, and all that. Okay, they are acoelomate. Okay, they are acoelomate. Then you have to remember that they have a water canal system which we just discussed. Oh, we forgot one thing, that is their skeletal system. Yes or no? The skeletal system is made up of, okay, we did not forget writing there in the before one, made up of spongin fibers, okay, made up of spongin fibers and or specules, okay, spongin fibers or specules, very important. Then what do we know about them? The sexes are not separate, okay, sexes are not separate. Fertilization, thank you. Okay, fertilization is internal and they have a larval stage. Okay, larval stage is present. This is how much you have to know about phylum Porifera. Okay, phylum Porifera, and then here you can remember about Ostia, Spongocele, and Osculum. Okay, and here remember about collar cells. Yes, did we miss something? Did we miss something? Tell fast. Okay, next we are going to Coelenterata. Okay, next we are going to Coelenterata. Okay, Coelenterata. Coelenterata, they are mostly aquatic. Students, Coelenterata has another word called as Nidaria as well. Okay, Coelenterata has another word called Nidaria. So if you see somewhere Nidaria, don't be like, oh, you ma'am, I don't know what is Nidaria. Coelenterata and Nidaria is the same. Okay, mostly marine, sessile, free living, radially symmetrical. The name Nidaria is derived from the word which you are going to see later. This one, all of you know, no, jellyfish. Jellyfish, all of you know, right? Now look here. This is the most important. Okay, the word that is nidaria came from the word called as nidoblast or nidocytes, which contain stinking capsules or nematocysts. That is very simple, students. If the coelentrates, no, they're organisms, no, inside that they will have a tube or thread-like structure. Okay, they'll have a thread-like structure which actually has a poisonous thing like this. A thread-like thing will be there. Okay, like this one thing will be there inside that. Now the moment they come in contact with a predator or a prey, okay, immediately they will release this stinking capsule, or they will release this nematocyst, and they will release the capsules and kill another prey or the predator. Okay, so that is the word. These are poisonous stinking glands, okay, stinking thread like structures which will come out immediately, roll over the prey, and they'll have stinking capsules all over this. Okay, nidoblast cells, okay, they'll have all stinking capsules all over them which will release toxins that will actually harm the prey or the predator. Now nidoblast are anchors. See, look here, they're going to explain more about it. See, nidoblast are anchors, are used for anchor defense or for capture of prey. This is what nidoblast is. Okay, nidoblast, you people can google and see how nidoblast looks like. You will understand it is like one thread like structure, and the thread like structure will go around and catch the prey. Okay, this nidoblast can be used for what? Holding, like anchoring to some place, for defense as well as for capturing of the prey. Nidarians exhibit tissue level of organization. There it was cellular level of organization in Porifera. Here it is tissue level of organization, and they are diploblastic as well. Okay, diploblastic. Now they have a central, very important, they have a central gastrovascular cavity with a single opening mouth on the hypostome. Okay, they have a single gastrovascular cavity with a single opening called the mouth. Okay, now digestion is extracellular and intracellular, that is happens outside and inside, okay, between the cells. Now some of the nidarians, example corals, have skeleton composed of calcium carbonate. All of you know corals. Okay, corals, all of you know, no, calcium carbonate. Nidarians exhibit two basic forms called as polyp and medusa. That means they have two body forms. It neither can be a polyp form which is attached or sedentary, or a medusa form which is like a jellyfish. Okay, now the former is sessile. This one, this polyp is sessile and cylindrical form like hydra, adamsia, etc., whereas the latter is umbrella. Latter means medusa is umbrella shaped, and free living like your Aurelia or jellyfish. I'll explain you detailedly this. Don't take tension. Those cnidarians which exist in both forms exhibit alternation of generation. That is called as metagenesis. That is, polyp will form medusa asexually, but medusa will form polyp sexually. Okay. Example: Physalia, Adamsia, all that. Wait. First, I will try to explain you what we just understood. So, students, they are telling coelant traits. Okay, coelant trait are tissue level of organization. Okay, tissue level of organization. Okay, then they are telling that corals, okay, corals, all that is there. Now see, I'm not going to go to this portion. Okay, this portion is not needed. I'll go to the later portions. Okay, later portions I'll go. Now most important, they have a, okay, they have a gastrovascular cavity. They have a gastrovascular cavity, okay, with a mouth, okay, with a mouth on them. So that means this is an incomplete digestion, only one opening is there, okay, and which is called as hypostome, which is called as hypostome. Clear? Yes or no? Clear? Now students, and remember this way, I think I can draw a structure and show you. This hypostome, no, is present near the mouth only, okay, near the mouth only this is present. Now how they will be present, you know, like this. Let's say I'm drawing this structure. Let's say I'm drawing a polyp. Okay, this is a polyp which will be attached downwards. Now this is the mouth. Okay, this is the mouth, a large mouth like this will be there. This hypostome is a region which is near to the mouth, a raised region. They will have tentacles around them. They'll have tentacles around them. All of you have seen hydra, if I'm not wrong. Like this, tentacles will be present, students, everywhere. Okay, 1 became 1 size, but this is how much I can draw with the pen. Okay, like this, tentacles will be present. Okay, so this portion is called as the hypostome. Okay, hypostome, they'll have tentacles. These tentacles are going to help them to actually catch the prey and all of that. Clear? Got it? Yes. Okay, now they told the next most important thing that they told was they have two life forms or body forms, right? Two body forms are there. Body forms are there. That is called as polyp and medusa. Polyp and medusa. Yes or no? That's what they told. Correct? Once again, yes. Okay. Polyp and medusa. Now this polyp and medusa, students, remember this polyp is actually a sessile one. Sessile means what? Attached one. Okay, this one. This is a polyp. Now medusa means what? Okay, medusa is going to look like this. Okay, like this. Got it? They will have tentacles downwards. Okay, they'll have tentacles downwards like this. They are free living. Okay, they are free living. They'll be happily living freely. Now they are telling that if this body forms can be reversed, that not reversed, like they can do metamorphosis. That is from one metagenesis, from one to another form they can form. That is, this is a polyp. This is a polyp. Okay, this is a medusa. This is a polyp. This is a medusa. Now remember that if it has to go from polyp to medusa, if a polyp has to form medusa, it will form it asexually. Asexually. But if medusa has to form a polyp, okay, if medusa has to form a polyp, if medusa has to form a polyp, it is going to happen sexually. It is going to happen sexually. Got it? If a polyp has to form medusa, it will be asexually. If a medusa has to form polyp, it is going to be sexually. Clear? Sexually. Okay. Now this is what they do. Okay, this is what they do. This polyp, students, is one body form, that is the form where it is attached. Then there is another body form which is floating, free living. Okay, free living or free floating. All of you hydrate yourself. Yes. Clear? Okay. This is your medusa and polyp. Polyp will form medusa sexually. Medusa will form polyp sexually. This is what they have told. Now look here. That is why it is called as metagenesis. Now umbrella shaped free living like Aurelia or jellyfish. Now example is going to be Physalia, okay, which is Portuguese man of war, Adamsia, okay, Pentalium, if you can remember, but at least remember PNA, okay, PNA, that is Physalia and Adamsia, okay, Physalia, Adamsia, that is Portuguese man of war, sea anemone, you can also remember jellyfish or Aurelia, okay, jellyfish or Aurelia. So three you can remember: Physalia, which is Portuguese man of war, you can remember Adamsia, you can remember Aurelia, okay, Aurelia or jellyfish. This three examples, at least three examples you have to know. Two examples is what usually NCERT or NTA tells, but you can go up to three examples because there's chances that you'll forget. Okay, now students, these people have tentacles. Always remember, these people have tentacles, and these tentacles are resultant. These tentacles will have your nidoblast cells. Okay, these tentacles will have your nidoblast cells. Don't forget that. These nidoblast cells will have poisonous stinking capsules. Okay, stinking capsules, and these stinking capsules can be called as nematocyte. Okay, nematocyst. Okay, sorry, nematocyst. These people are going to help in capturing the prey or harming someone. Okay, done. This is how the word term nidaria comes for them. Okay, nidaria comes for them. So this is the example. See, look, this is a medusa form. Okay, this is a medusa. Okay, this is also a medusa. So if a medusa has to become polyp, it will do it sexually. Okay, sexually. If a polyp has to form a medusa, it will form it asexually. Okay, asexually. Now this is going to be a polyp form which is sessile. Okay, and here can you see, this is the hypostome region. Here, this region will be the hypostome where the tentacles are all present. Okay, and they will have a gastrovascular cavity as well. This is your medusa region where the tentacles are here. In this tentacles, always remember nidoblast cells will be there. Okay, which are going to stink. Okay, which are going to stink. Done? Clear? Okay, next is Ctenophora. Okay, next is Ctenophora. Let's see what is Ctenophora. Ctenophora is actually very easy. Okay, very small kutti paragraph Ctenophora is. Ctenophora, commonly known as sea walnuts or comb jellies, are exclusively marine, radially symmetrical, diploblastic organisms. Okay, what are they? They are marine, radially symmetrical. What were these people, students? Did we forget that to write it here? What they were also radially symmetrical. Okay, that also we will make a note. That is, if we cut them in any plane, they will still have the same symmetry. So these people also were radially symmetrical. This is very important. They'll give you a box and they'll ask, identify the ones which are radially symmetrical, identify the ones which are bilaterally symmetrical. Okay, this fellow also is radially symmetrical. Okay, radially symmetrical. Radially symmetrical. Now they are diploblastic. Okay, diploblastic. What you have to do now when you're learning this chapter, make a paper and write all the bilaterally symmetrical, all the radially symmetrical, all the diploblast, all the triploblast. Make a column and write. Otherwise, this chapter you will not understand very easily. Like in one go, you cannot. Like you will understand in one go, you will not be able to memorize all of it. Okay. Diploblastic organism with tissue level of organization. The body bears 8 external rows of ciliated comb plates. So always remember there are going to be some general characters. right? Like tissue level organization, cellular level organization, triploblastic, radial symmetry, bilateral symmetry. Those are general characters. Here we are talking about specialized characters. Okay, here we are going to talk about specialized character. Here you have 8 external rows of comb plates which help in locomotion. Digestion is both extracellular and intracellular. Okay, same thing like your tenophores only. Now, bioluminescence. This is a very important characteristics. Bioluminescence, that is a property of living organism to emit light. Okay, is well marked in tenophores. Sexes are not separate. Okay, sexes are not separate. Reproduction takes place only by sexual means. Fertilization is external with indirect development. These things are not major consideration. Major consideration is their specialized characters. Okay, major consideration is the specialized characters. So here students, remember your tenophore, no? It looks something like this. Okay, like this, with like this. Okay, like a football with hair. Okay, football with hair it will look. Okay, like this it will look. Like this. Okay. Now what are they telling? These ones are radially symmetrical. Okay, and they are diploblastic. Okay, they're diploblastic. Now, most important thing, they have 8 ciliated comb plates. Okay, 8 ciliated comb plates. What is this comb plates going to do? Locomotion. Okay, 8 ciliated comb plates are going to do locomotion. Clear? Then they are telling that they have a most one of the most another feature is that they show bioluminescence. Okay, bioluminescence, where they will be able to emit light. Okay, emit light. Other things are general characters like sexes are not separate. Okay, sexes are not separate. Fertilization is external. Okay, fertilization is external. Then they have indirect development. Indirect development. Got it? These are all very common characteristics. What you need to focus mainly is going to be about these two characters. Okay, 8 comb plate and bioluminescence. Clear? Example students, very easy. Tenoplana. Okay, tenoplana and plurobranchia. These are the examples you can remember. Tenophora means tenoplana will be easy to remember. Okay, so here you can write plurobranchia, branchiae, and tenoplana. Okay, tenoplana. These are the two examples. These are the two examples. Clear? Yes or no? Clear? Okay, done. Okay, this is your tenophores. So students, before platyhelminthes, we'll take a break. Okay, I'll be back. So I'll write it here. Okay, here, yes. By this time, I'll try to fix the mic as well. So just give me 1 minute.